Okay, perfect. Yes, thank you, Anais. It's a really great introduction. As I said, it's I've been very lucky having the opportunity to work on a very constant problem throughout many different phases of my academic and then scientific career in general. So as you pointed out, yeah, so my main interest that I've always been extremely focused on is to understand how biological systems are organized using genotype to phenotype connections as a main tool. Many of us are attacking this problem for a variety of different angles. In most recent years, the ones that come to mind the most are genome-wide association studies that try to exploit the genetic variation in natural human populations that are data for which is becoming online more and more quickly. So these are GWAS and PHEWAS analysis. At the same time, breakthrough discoveries in functional genomics technology in mammalian cell lines have enabled cancer vulnerability screens and other similar approaches where you knock out, eliminate, or create a particular genetic variation in a cell line in a controlled environment, a controlled genetic background, and then look for phenotypes associated with that. Generally speaking, they're fitness defect searches in an attempt to understand what makes cancers vulnerable to specific perturbations. Also, most recently, there's a whole other area that's becoming more and more exciting to me, and that is the search and the analysis of naturally occurring human knockouts. So these are individuals that, for a variety of historical and genealogical reasons, happen to be carrying protein truncating variants or other inactivating mutations, either as heterozygotes or homozygotes, and as a result manifest or maybe don't manifest certain phenotypes that one would expect. So as more data becomes available through large-scale sequencing of bottleneck populations or highly inbred populations, this data will also become more and more important. All this taken into account, we shouldn't forget that the area where the most data and the most understanding about genotype to phenotype connections has been developed is in model organisms. And that is partially for historical reasons. They became amenable to genetic perturbations much easier, and as a result, the number of tools is hugely abundant in model organism compared to mammalian cell culture, human cell culture, or human populations. And so for all of those reasons, we have more data and we have better understanding of those systems. And everything I'm going to tell you today will exploit this particular aspect of model organism genetics and phenotypic analysis in yeast. But I want you to keep in mind that nothing of what I say is specific to yeast. All the modeling, all the logical and biological interpretations of the results and of the connectivity between genotype and phenotypes will be relevant, is already relevant in other systems. And so just a matter of deciding, it's just a matter of waiting when the data becomes available and we can do the same. So just to be clear, what we're talking about, what I'm going to be describing is all the data we have today and the analysis we're doing, trying to understand the relationship between targeted perturbations in unicellular eukaryote, which is Yeast, Saccharomyces cerevisiae, and a variety of phenotypes that have been collected for that. So just to give you a little background, yeast has been the first and one of the few to this day organisms that have has a knockout library available to it. About 20 years ago, the entire yeast community got together and developed a plan and executed a plan to knock out every single open reading frame in the yeast genome. So that's about 6,000 genes. Every single ORF has been replaced with selectable marker flanked by two unique gene-specific molecular barcodes. And this entire library of mutants became available to the entire community in a variety of formats, the most popular of which is just a set of 96 well plates where every well contains an isogenic population of yeast cells, each of which is knocked out for that same gene. And then the neighboring well will have a different mutation. So one of the first observations that was made with this library, or in the process of making this library, is that about 82% of yeast genes could be knocked out with no major consequence to the cell. The flip side of it is that 18% were considered essential. So these are genes that, when eliminated from the yeast genome, were not compatible with viability in standard laboratory conditions. So the reason the interpretation of why 82% were not required for viability, there were many different interpretations. The field kind of divided into two different directions and followed them in parallel. On one end, one interpretation was that the function, it's not that these 82% of genes were not important, is that their function is not required under those particular conditions where the strains were being made. And that is rich medium where yeast doesn't have to produce anything, all the nutrients are provided to it. And for a phenotype such as growth, which is one of the many things that one would want to investigate for a knockout. And so the decision there to, in an attempt to explore this, the decision was to try to understand what other phenotypes and what other environments might uncover the requirements for all those particular mutants that did not show a fitness defect. In parallel to that, and I'm going to come back to this in a few slides, the other interpretation was that the function of these genes was extremely important for viability as well, or at least a huge fraction of them. But the function was so important that evolutionary, the yeast developed a buffering system where the function of those genes were buffered, were compensated by the presence of other genes. And so the one way to investigate that is to try to create higher order combinations of knockouts, for example, double mutants, trying to uncover such redundancy. So I'm going to talk about both of those directions that the field took. I was lucky enough to be involved in both of those investigations. And I'm going to try to show you how it's the union of the two, the integration of the two data sets, or the two approaches that brings the most power to yeast as a genetic system. So first of all, other environment and phenotypes. So in the 20 years since the release of the deletion collection, about 400 papers have been published in the literature from about 300 labs and counting. The number still keeps increasing, where every paper usually contains a screen or a number of screens where the entire deletion collection has been tested for something. Here's just a sample of the phenotypes that were tested for all non-essential deletion mutants. So the phenotypes vary from morphological features such as size, shape, form, budding patterns, and so on. Subcellular phenotypes, shape, number, and distribution of certain organelles, or in this case, lipid droplets. Abundance of certain amino acids or other metabolites. Gene expression profiling on a very large scale. Stability of DNA and mRNA species. A number of reporter screens have been developed to try to assess the efficiency of certain molecular pathways, such as transport or modification. Usually it involves a fluorescently marked protein that is involved in that process, and you just follow the behavior of every single deletion mutant in that direction. Up to the point that the deletion collection was sent to space at some point to measure the effect of microgravity on all the deletion mutants. That's an interesting study over there. And obviously the most frequent and the most abundant data set is testing growth under a variety of stressors. So a lot of them are drugs, both known and unknown. Some of them don't even have a name, they only have a chemical structure, but also nutrient limitations, temperature shifts, osmotic stress, anything that scientists thought could have an impact on the growth of the deletion mutants. And by looking at differential phenotypes across the mutations, you might be able to figure out both what the genes do and what the perturbation does. So when I started thinking about it, this was a few years ago, I quickly realized, and I think it becomes obvious when you think about this in these terms, is that this is the single largest, richest, and most systematic phenotypic description of an organism. There is no other species on the planet that has been this thoroughly examined by looking at thousands of phenotypes, hundreds and thousands of phenotypes in isogenic populations missing one gene at a time. Obviously it has enormous potential in my mind for understanding how genes are related to one another and how the whole system works. However, this richness, this rich data set, or this rich resource has never been analyzed in such a way. And the reason for that is because I often say having a lot of data is not the same as having a data set. Every single piece of data that I'm showing you here is scattered throughout the literature. Best case scenario, it's an Excel file in the supplementary material of a paper. Worst case scenario, it's a screenshot of a table saved as a PDF in a supplementary material. I guess the worst case scenario, that's no data at all. It can be recovered, but those are rare cases. So in order to analyze this data, it was obvious that some data gathering and organization needed to be done. And after a little bit of reflecting, I decided that I care enough about this problem to take on that endeavor and start what now we refer to as the Yeast Phenome Project, whose main goal is to collect, organize, and analyze all the systematic screens, phenotypic screens of the yeast deletion collection. Very briefly, what that entails is reading every single one of the 400 papers that I mentioned, identify the data sets within each paper. Each one data set corresponds to one genome-wide screen. Annotate which phenotypes and in which environment, what was measured, and annotate both of them using a controlled vocabulary so that you can draw connections between similar phenotypes and similar environments. So you can then group data sets based on your question of interest. Retrieve the data, which often means talking to the authors, because there is more data that is published sometimes, and the authors are usually very kind and decide to share with us even versions of their data sets that are more complete than what was originally published. And then basically do some basic cleaning, restructuring, reformatting, all that to make the data comparable to one another and make it analyzable. I want to emphasize that early on we decided to be avoid manual manipulation as much as possible. Everything we do, starting from, let's say, an Excel spreadsheet that downloaded from a supplement, is cleaned up and reformatted computationally, and the code is version controlled and saved for future editing, potentially, if one decides to change your mind how to do it. And everything gets stored on GitHub. It's in a private repository for now, just because it's work in progress. Eventually, we're just going to open up everything and everybody will see this history of what happened to the data and how we decided to manipulate it relative to the original. And as well as onto our website, which is not quite ready for prime time yet. It's kind of a draft form right now, but we are very serious about making this as useful and as practical as possible for everybody who might want to use this. To give you a sense, we're like constantly at 90% done, but partially because we're making progress, annotating and downloading more data, but also the space that we need to cover slowly increases, so we can never reach 100%, but we're in a very good point that allows us to have data for 6,700 phenotypes and 5,000 environments, for a total of 15,000 data sets total, 15,000 genome-wide screens of the deletion collection. I wanted to mention that I can talk for hours about the process of putting this data together. It's been a very interesting experience that taught me a lot about how we produce data, how we decide to store it, what do we do with it. And I put some of those thoughts on paper in a recent opinion piece that I wrote for the FABS journal. If you're interested, I would like to point you to that short manuscript that kind of describes the experience, and more abstractly, what would I recommend going forward in terms of similar projects. Okay, so basically what we currently have, and this is a set of data that we have been working on, is a matrix of about 5,000 rows, each corresponding to a knockout mutant of a non-essential gene, and about 11,000 columns in this case. These are all the screens that employed either the haploid version of the deletion collection or homozygous diploid. There's also the heterozygous diploid that needs to be analyzed separately, and so we're not looking at it right now. As I'm showing you here, about half of the data, the blue part, is growth in different environments. Those are the easiest, the most popular experiments. Another half, which is not complete in this case, the yellow part, is a gene expression data set. So only 1,500 knockout mutants have been profiled for gene expression. So it's not technically a genome-wide data set, but we decided to include it anyway because of its value and uniqueness that will help understand the other phenotypes as well. And then there is this purple-pink slice in the middle, which started very thin, but it's growing pretty substantially over time. And these are what we call other phenotypes. These are all the unique and very difficult to measure things, such as morphology or efficiency of pathway, or certain pathways of transport, of distribution of organelles and other things, abundance of certain metabolites that are much harder to measure on a large scale, and that makes them so much more valuable. Also, these data sets tend to be published in smaller papers, and as a result, they have really never been analyzed in the context of other data sets. While chemical genomic screens usually come in big data sets, they have been analyzed together or integrated with other data sets. These other phenotypes have not been. So in my opinion, they are the most valuable going forward. So having so many data, at least at a certain point in our aggregation process, it is normal to ask, like, what now? What are the first questions you should ask having collected such data? There's 1,000 different questions that we had on our plate. We decided to stick to 3 major ones and a fourth that kind of serendipitously we came across. I'm not sure I'm going to have time to touch upon that, but I will mention it at the end if I have. But the 3 main questions are kind of the base for everything else one wants to ask of this data. Number 1 is data quality and reproducibility. As you collect the data, you realize a lot of screens have been done multiple times by independent authors over like decade apart from one another. So a common question is how similar are the results and how much we should trust every single one of those screens. The second question is how good is this data to infer relationship between genes? Not only obviously just associations, like which genes are likely to share common functions, but also potentially, and this is something we're exploring at the moment, can we tackle the not only abstract associations, but also maybe potentially ordering in pathways that has been kind of a difficult problem to tackle historically. Same thing, number 3 direction is infer phenotype-phenotype relationship, including, similarly to genes, causality relationships, not only understanding which phenotypes tend to co-occur in the same mutants and what does it mean about biology of yeast, but also can we try to figure out which phenotype is causal of the other phenotype. This comes from my mild exposure or moderate exposure to human genetics at Calico, which is a very well-developed field where those problems are central to understanding the roles of genes in disease, but also the roles of intermediate phenotypes in causing or following a disease. So all of those questions are very relevant, not only to yeast, but to all other molecular systems and biological systems as well. So let me tell you a little bit what we have been thinking about in terms of data reproducibility. As I said, there's many experiments have been done multiple times. Here I'm showing you just one example of such an experiment. It is a search, typical search for respiration deficient mutants. So yeast typically grows on glucose very nicely. It can also grow, generally wild type yeast can grow on other non-fermentable sources, carbon sources. but mutants that are defective in respiration cannot grow on those media. And so it has been an experiment to identify all respiration deficient mutants there, which is mutants incapable of growing on non-formable carbon source, has been done many times. We could detect at least 5 times by 4 different labs over a 10-year period. Each experiment is a genome-wide experiment. They tested 5,000 mutants each. They each identified about 300 hits, about 6% of the genome, which is interesting that the hit rate was similar. When you look at the overlap between those hits, the picture is also not too bad. Here I'm showing you for each of the 5 experiments. I'm showing you it's a form of Venn diagram of all the hits in this experiment, how many of those also were detected in all the other studies, 3 out of the 4 studies, 2 out of the other studies, and so on. So if you focus on the red and the yellow sections of each bar, you will notice that they all hovered about 60%, meaning for every single experiment in this set, 60% of their hits were recovered by at least 3 of the other 4 studies, or in total 4 out of 5, which is not bad at all. But it raises the question of what's going on with the other 40%. Are they false positives in each individual screen? Or are they false negatives in the screens that they did not appear? And most importantly, how can we assess, how can we answer this question when we don't have a reliable ground truth? And we don't have one because these screens were supposed to be the ground truth. We were hoping that by screening knockout mutants on a non-formable carbon source, we will finally have a definite list of respiration deficient mutants. There's no other way to detect it. So it turns out it's not as easy and we don't have a ground truth. So how do we answer that question? Here I'm going to take a little digression and go back to this point where I told you that while one part of the yeast community was screening the knockout collection for different phenotypes in different environment, another smaller part of the community was focusing on testing higher order combinations of deletion mutants. And this will become important to help interpret the knockout screens, such as respiration deficiency. So what does it mean to test higher order combinations? One of the reasons to do that is to detect genetic interactions, which are unexpected phenotypes that would not have been predicted by the phenotypes of the single mutant. Primary goal is to detect synthetic lethality, which are cases where 2 single knockouts don't have a visible growth defect, but the combination of both in the same cell leads to death or inviability. This is interesting because one of the explanations, not the only one, but one of the explanations is that those 2 genes would act in parallel pathways that all come down to an essential function. There's also other type of interactions you can detect from looking at single and doubles together. One of them is you can potentially understand which genes work in the same pathway by looking at the phenotype of the double mutant that is not substantially different from the phenotype of the singles, even if you do expect the double to be a little sicker, for sure. So you can measure this quantitatively. Obviously, there's a huge field on this topic. This was part of the work I've done a few years ago while first being in Charlie Boone's and Brenda Anders and the whole team in their labs and working with them, and also continuing that while I was at Princeton. This huge collaborative effort led to the creation of a gigantic data set that covered about 20 million mutant pairs, which is a substantial fraction of all possible combinations of deletion mutants, and not only deletion mutants in yeast, identified about 1 million genetic interactions and so on. It's a huge data set that will be analyzed for many, many years to come, and we are for sure doing that. I want to be synthetic, want to be brief, and I want to focus your attention on the 2 most important lessons that we learned from that data, in my opinion. The number 1 lesson is that strong genetic interactions, such as synthetic lethality, which was kind of the main motivation or the primary motivation for doing this experiment, those interactions are extremely rare. They're less, depending how you measure them or how you count them, it's less than 2, maybe less than 1% of all the tested pairs. However, when they are, when they do find them, when they do occur, they're extremely informative about the relationships of the genes they involve, just like a parallel pathway would be. The second most important lesson is that sharing genetic interactions, not just having a direct connection between two genes, but if two genes share similar interactions with other genes, no matter whether it's strong or weak, it's even more informative than having an interaction itself. And this plot, it kind of captures a little bit of what I mean by that. If you do a precision recall analysis on either positive interactions, negative interactions, which are epistatic and synthetic lethality interactions in brief, they have a certain ability to recover known gene pairs that are co-annotated to the same Go term. The black line, which is the correlations, the correlations of their genetic interaction profiles is much more accurate in recovering those known pairs. This property of genetic interactions enabled us to make these kind of networks, which are genetic interaction similarity networks. They're correlation networks. They summarize millions and millions of different data points and basically connect gene pairs that share interactions with one another rather than having a direct interaction themselves. You might have seen talks on this topic in other situations where we showed through a variety of different techniques that if you embed this network in 2D using one of the many available tools, you end up with very large domains or very large clusters of genes that all share similar functions with one another. And it's much deeper than just saying, if you cluster this network, you obtain groups of genes that share functions. This function distribution seems to be much more continuous, much more hierarchical. The definition of function itself needs to be reconsidered, I think, when one looks at the data this way. And all of that needs to be investigated much, for many years to come. On the right here, I'm showing you a simple approach that enables you to run functional enrichment on all of the different areas of the network. And you can show that all these major clusters, major areas of the network correspond to a known function. And that's your anchor. You can recapitulate known biology by clustering genes this way, but there's a lot more than known biology in here. And I'm gonna touch upon this as well. So one thing that this network enables you to do once you know that it recovers known biology is that you can use the network as a benchmark to evaluate data that you know nothing about. One strategy to do that is to calculate local enrichment for phenotypes of interest or for properties of interest that are associated with genes as they are distributed throughout the network. Again, there's many approaches to do that. We adopted a simple one where you can basically take one node at a time on the network, define its local neighborhood in a variety of ways, either a certain number of hops away or a certain distance away if you want to exploit the length of the edges that result from the embedding. And then once you define the neighborhood and you define a score for that neighborhood, you can compare that to randomized versions of the network and basically decide whether the neighborhood is enriched for a particular property. And then as you repeat it throughout the entire network, you can get a general sense of which part of the network is enriched for a phenotype or property of interest and which ones aren't. The reason I'm telling you all that is because if you go back to this original question, which is respiration deficient mutants and how reproducible they are, I told you only 60% of the mutants are reproduced across different screens and the other 40% are not. You can take each of those data sets, overlay them onto the network, and ask what is the general trend of what are the different areas of the network where each of those hits lists are enriched in. And the answer is that the enrichment profiles of the different data sets are nearly identical. They're basically the same thing. So what they have in common is probably real, and what it's different between them is probably random, it's probably noise, because it's scattered throughout the network and not consistent across in any particular domain. One way to look at that is, on the left, I'm showing you enrichment profiles where the darker the color, the more enriched that neighborhood is for hits from any given screens. I'm showing you 3 different screens here. You can also plot the results of any particular screens and color the nodes based on how reproducible they are. And you will notice that the ones that are reproducible, the red and the yellow ones, are the ones that correspond to the only region pretty much that is enriched for hits in any given screen. So if you didn't have 5 replicates for a particular screen, but only had 1 and mapped it onto the network, you would basically get the same answer. So I hope that makes sense. So in the interest of time, I'm going to start, actually almost should wrap it up, but I'm going to touch upon what other things we can do. So infer gene-gene relationships, it all comes down to, if you compare phenotypes from thousands of phenotypes across many different genes, you recover genes that are known or that have not been characterized before, but you can predict function for. A good example is this one guy, YHR045W, which has no known function, which is bizarre because in this particular data set where I'm looking at genome-wide screens for sensitivity to hydroxyurea, which is a very well-characterized drug, you will notice that, so on this graph, the x-axis corresponds to rank, and every bubble corresponds to a different screen. So this gene YHR045W came up in the top 10 hits of the 15 screens that have been done so far. It was in the top 10 hits for half of the screens, and it has never been mentioned in any of those papers. Nobody thought it was worth following up on it. We were curious about this, digged out some data that was known about it. So physical interactions was the only thing that it had of other data sets. We noticed that of the 4 interactors that have DAP1 sounded familiar, and that's because it was right here next to it, had the same phenotypes. So we investigated this a little further. Basically, long story short, we found that YHR and DAP1 are what we call each other's best buddies. They share more phenotypes with one another than with any other gene in the genome, and the phenotypes they share are, have been associated with DAP1, and there's a molecular explanation for that. So we are very confident that YHR probably works with DAP1 in some way, and we can confirm that the phenotypes are real. They are driven by the mutations themselves and not by any other possible perturbations associated with knocking out these genes. And we're currently in the process of figuring out what is the relationship between those 2 genes. I want to point out that even if you can do it on a gene-by-gene basis, it's not sufficient because there is 1,000 unknown genes to this day in yeast. And I'm showing you here on the map how they are distributed. Some of them fall into clusters, so you can probably guess what the function is from there. But many of them are scattered throughout the network, so you may not be able to do that. However, in 83% of these genes have at least 1 strong phenotype in yeast phenome, and 63% of them are strongly correlated, just like YHR is, to at least 1 characterized gene. So there seems to be potential here to finally at least name them, give them some name that is indicative of their function. And we've decided that we're going to take this on and tackle it. Also, I want to point out that in addition to, I mentioned this earlier, that while this network recapitulates a lot of known biology, there is still a lot of things we don't understand. One of those things, turns out there's a little cluster in the network that have not been annotated biologically before because it doesn't have any known association with it. Go doesn't know anything about these genes. It is enriched, however, it's composed almost entirely by uncharacterized genes. And based on the phenotypes that they have, normally we would ignore it because, you know, it's probably noise. It's unlikely that there's still a completely uncharacterized function in yeast. But because of the yeast phenome data we collected and because of the phenotypes that all of those uncharacterized mutants share, we now can formulate the hypothesis that this actually might be a functional unit on its own. And we're going to try to tackle it and try and understand if we can pin it down. All right, very quickly, phenotype-phenotype relationships. It all comes down to the fact that similar to genes, some phenotypes are not, if you compare 2 phenotypes, like in this case, lifespan and cell size, that have been hypothesized to be related, if you look at gene-by-gene comparison, there doesn't seem to be a strong relationship between them. But if you map them onto the network, you will find out that there is, as predicted, there is a strong relationship, in this case, an anticorrelation between mutants that are small and which functions they are enriched for, and mutants that are long-lived, and vice versa. And this helps us think about what causes longevity or short lifespans, and what are the potential molecular mechanisms that might be underlying it. And this is just some additional data we have collected. So in the interest of time, I'm going to skip a couple of other things I wanted to say, and just finish on saying that, as you can tell, what we're interested in is an integrative analysis of genome-wide phenotypic screens. We have collected them because we believe that the union of such data is much more powerful than each individual data set separately. No matter how big they are on their own, the union is always better. We can evaluate reproducibility, we can assign new gene functions, we can identify related phenotypes on a much larger scale that I had time to show you. And most importantly, this process of aggregating, annotating data sets is what I prefer to call it a data librarianship. It's much more than just sharing data. It's making data usable for people who did not produce it, such as us in this case. And I think that is essential going forward for many other similar experiments. So on that, I want to thank, obviously, the team, my team at Calico and my collaborators at Calico who helped me think through this. Some of the folks who I started working at Princeton and continue to do so was super helpful to making sense of the data. Also, a huge thank you to the Yeast community. As I mentioned, we interact with the authors a lot. We talk to them all the time, trying to make the best use of the data they made available. And a big shout out goes to them for making this possible. And thank you so much for listening. I'm happy to answer questions.
Many thanks, Anastasia, many thanks. In fact, I have to say that working in human genetics, I'm feeling really jealous to see how people are willing to share the data in the Yeast community, because we are really struggling with that. So there is a high-level question from Tuyana. Calico seems to be interested in studying aging. So she's asking if you are directly involved in the aging-related efforts. And can you comment on where aging research is and what are key challenges to promoting longer, healthier life?
Well, yeah, it's a much bigger question. So Calico has a unique interest in understanding the basic biology of aging. As you can imagine, that's a very, very ambitious goal, and it will take many years to make breakthrough discoveries in that. So in that sense, I don't really have a kind of a quick answer of like what are the next steps that Calico is taking. We're taking many different steps. We're studying yeast, a replicative lifespan in yeast as a model for what aging might look like in humans. We study naked mole rats, we study mice physiology. It's a very, very broad effort. And I'm partially involved in some of those things, but the benefit of being in Calico is that I can study, take a deep dive in yeast biology in an attempt to understand how yeast works. So we can maybe understand how yeast ages so that we can maybe understand how humans age. So it's a very long path, but necessary path to take. And I think I'm very lucky, and all of us are very lucky to have the time and the dedication to take that path. Oh, you're muted, I think. Yeah.
Yes, indeed. Sorry, so another question from Lenore Cohen. She's asking how well do sequence paralogs cluster in the similar genetic interaction space that you mentioned?
Sequence paralogs in genetic interaction space?
So, yeah.
Yeah, so paralogs are kind of a little bit tricky to study this way, because in principle, single mutant, so if you make a single knockout of a paralog, if the other paralog is there, the genetic interactions will be weak because the other paralog is always there to compensate for it. It's the same with single mutant phenotypes. Paralogs are buffered by the other paralog. So they do tend to have synthetic lethality interactions more often. That is true for sure. But in terms of profiles of genetic interactions, as far as I know, they're not that powerful because of the buffering. So that makes them a little harder to study. But there have been a few papers even recently that have tried to disentangle that and try to what we can say by comparing the genetic interaction profiles of two paralogs and try to understand what functions they share versus what functions they differ in.